Welcome, everybody. Today we are very excited to have our March seminar and meetup. A forward. Those of us, or maybe only me in the United States, it's a little early here, so if I slip up and say something silly, it's 5 a.m. You cannot hold me accountable. Today we will have 3 speakers, and we'll all be talking about the theme of continual learning, evaluations, and applications. So I'm very excited. We're going to have some of the talks, especially the second 2, will be very different than what we're used to having here, and I think that will be great for all of us to pay attention and listen to. Each speaker is going to have 20 minutes total for talks and questions, and then after all 3 have gone, whoever can stay, we will open up to additional questions and general discussion. And as a reminder, we are very excited to have this new and more interactive format. We are open to tweaking it in the future, so if you have any feedback or speaker suggestions, please reach out at any time. So we're going to start off with Matthias de Lange. Sorry, it's very early. I'll first say he just got his PhD, so we'll have to give him a round of applause. He defended yesterday, so now he is a PhD graduate from KU Leuven. He'll be speaking about his iClear 2023 spotlight paper. You may be familiar with him. He is actually one of the co-chairs and organizers of the CL Vision workshop. And I'll go ahead and turn it over to you, Matthias. We are very excited to hear this paper.
Yeah, thank you so much, James, for the invite and for the kind introduction. Today I'll be talking about continual evaluation for lifelong learning and how we identify the stability gap. So in my PhD, I've mainly been working on continual learning, so really the learning part. But the question I was really intrigued with in my last year is, don't we also need continual evaluation if we do continual learning, especially if we want to know what is going on during the learning process for a learner? So this work was in collaboration with Guido van de Ven and with my promoter, Tine Tertelaars. And so the idea is basically the trends in continual learning. We have a lot of work going on in online continual learning and streaming learning, where the idea is that you have an ongoing stream of data where the specific distribution shifts are not particularly known in stream. But there's also a lot of applications now occurring in continual learning, but I often see them also occurring in application areas where it's really safety critical. So you could think of self-driving cars, for example, also in the medical sector. And also there, like this continual evaluation or rethinking how we evaluate our continual learners could be really of importance. And actually, many of the main metrics that we have at the moment are often still based from the task-based era, like the average accuracy we have over the different tasks we're getting, backward transfer. And to compare to previous works, we need to often report the same metrics, but maybe it's time to also rethink how to evaluate our. So the first thing I was interested in is if we have like an online continual learner or you have a system that changes all the time, aren't you interested also in the worst case performance? So basically in our metrics, in continual learning, we're interested in 2 parts. On the one hand, we have the elasticity. On the other hand, we have the stability. And these 2 components, we want to measure them in some way. So the first one we look at is the minimum accuracy. We basically look at each task, and we have like evaluation tasks, and we look for each of them, what is the minimum accuracy we had after training that specific task. But the minimum accuracy will only look at our previous task, so it's more like worst case metric for our stability. We can also take into account the accuracy of the current task we are learning, and getting to our metric we propose, the worst case accuracy. So it's basically a combination of our minimum accuracy of our previous tasks and the current task we are learning. And then also, which is also a metric that makes it possible to measure the forgetting without having the need for these task transitions, is we basically look at a window of our preceding accuracies or preceding updates we had in our data stream. And within this window, we look at the maximum forgetting or the delta we have within this window. So with these worst case performance metrics, if we want to do a continual evaluation, we basically want to evaluate our model on every update, maybe we have on our continual learner. But it comes, of course, with some computational expense. But we also look at several ways we can make it more computationally feasible. And we see, for example, instead of evaluating on the entire set, we can just use subsets of, for example, 1,000 samples that seem to give a pretty accurate measure. Now, let's look at an example. So we have the typical class incremental split MNIST. So each task consists of 2 digits and try to classify. And we look at the accuracy of the first task. So usually with our task-based evaluation, this is what it would look like. And learning with replay, we see that there is actually no catastrophic forgetting, right? So replay is seems to be very effective. But we can now also try to increase like the periodicity of our evaluation. And for example, we could evaluate every 100 updates on our model. We still see a kind of similar trend. And going even further, when we evaluate on every single update of our model, we kind of see another interesting phenomenon. We see like this sharp drops on the task transitions, and it seems to be followed by some kind of recovery. Now, we not only find this for the split MNIST scenario, but we also find this on split cipher 10, split mini ImageNet in the class incremental scenarios. So split mini ImageNet has like 20 tasks, and we even find it for mini domainnet. So there's a domain incremental scenario where we actually have one output head with very sharp distribution shifts in the domain. And even here, we can also observe this kind of behavior. So also looking at our metrics for mini ImageNet here, for example, we would have about 33% average accuracy. So we would think like, okay, we have a very good performance. But if we now look at our evaluation periodicity, so for example, the first row, looking at our worst case accuracy or our minimum accuracy, you see that our minimum accuracy is like 0.5 accuracy for our previous tasks. So this means that on average, our previous tasks will drop at some point to like almost 0 accuracy. And this could also be the indication of the stability gap occurring, or this phenomenon that we saw of this performance dropping and recovering. And that's also what we refer to as this phenomenon, is that we have this transience, but still significant forgetting. But also part of it is the recovering process. So we basically have to forget on the task transition and the recover phase upon learning new experiences. Now, why is this important, this observation? As I mentioned earlier, we have this safety critical applications. Now, especially if we look and if we're interested in this worst case performance, like if you have a self-driving car, you don't want to go at some point going to zero accuracy and then recovering its performance afterwards, right? So for using continual learning in practice, it could actually be really important to look at this worst case performance, but also to prevent this kind of stability gap. But we could also think of an agent that is in control of the data stream. It could actually exploit this stability gap and make like, if the agent can control the training stream, it could like induce a stability gap by imposing a distribution shift. So there's also a point to think about. Also, something that's really interesting is, we observe this forget and recovering phenomenon, but wouldn't it be more efficient for a neural network to actually not forget and, just more efficiently relearn the new things that we're trying to learn in our new tasks? And we also think is a pretty interesting on the scientific level, is it gives some more insights in how replay works, where we thought like it just prevents catastrophic forgetting, but we see that it's actually this forget and recovering mechanism, and gives some more intuition how replays works. And it also poses the question more to the biological part, is do humans also suffer from this, transient forgetting if we learn something new? So there's a lot of open questions here that make it very interesting. Now there's a question that we also looked at ourselves, and that's how important is the task similarity and what is the effect on the stability gap? So we basically make a controlled experiment with rotated MNIST. So this means we have the MNIST data set for 3 tasks. But for each task, we make like a rotation on the entire data set. So we have this domain shift, and we can see if we increase the angle of this rotation and hence increase the domain shift, actually the stability gap also increases. So if we look at the red line, for example, which has the biggest domain shift, we also see there's a larger stability gap. Now, besides that, and I also think that's very interesting, is that it is also observable in the task incremental scenario, although from our results, it's less obvious than for the class incremental, which is also a much harder scenario. But we also can find it for we have the learning without forgetting and synaptic intelligence, elastic weight consolidation. And on these task transitions, we still can find this, even though it can be a small dip in forgetting and later this recovery happening. So this is also on the split MNIST data set. Now, we also provide a conceptual analysis of why this phenomenon might actually be occurring. And I like to think of it as splitting our loss and our gradients in 2 components. On the one hand, we have our plasticity. We want to learn our new task as fast as possible. But on the other hand, we have the stability component in the gradient, which is basically trying to keep our solution stable, and which is basically the part that's introduced by our continual learner, or continual learning mechanism. So basically between these 2 parts, it's like a tug of war, right? One hand, you want to learn the new things as quick as possible, but the other one wants to stay stable. And what we actually find with analyzing these methods in this way is that, for example, in replay, we find that the norm of the stability component is actually close to 0, because after you learn the task, you're basically converged to learning the task. And on this learning a new task, using the samples, you will basically have near to 0 norm gradients. And hence, you don't have the stability component, and it's just like you're basically fine-tuning. You only have the plasticity components. And hence, this could be a possible explanation of why we observe this behavior in replay. But we also found it right for the learning without forgetting and elastic weight consolidation using regularization. And here it's even more clear, because on these task transitions, what they basically do, in the regularization objective, you say you want to stay close to the solution of your previous task. But at that point, especially like your first update, that solution is exactly the same. So the regularization objective becomes 0. You have also a 0 norm gradient component. You can also observe this in the plots below for learning without forgetting. So without a stability component, you just have the plasticity component, which is basically fine-tuning and overwriting everything with the knowledge of your new task. So that was basically all of our findings and few insights maybe on the stability gap. And I want to thank you very much for your attention, and I'm happy to give answers to any questions. We also have the code available, by the way, so if you're interested on working on this, you can start from there.
Awesome. Thank you so much. And that was perfectly on time. So you must have had a stopwatch or something going. Great. So we are open now for questions. We'll take a few. And I see they're already coming. Okay. I'll first ask a question from chat, and then we'll go by hands raised. By minimum accuracy, do you mean minimum instance accuracy? I'm not sure if that question makes sense to you.
Minimum instance accuracy, or?
Yes, it's in the chat if you can see it.
Yeah, I'm not sure what you mean with instant accuracy, but indeed with the minimum accuracy, we evaluate on the periodicity of every update. So basically after learning a task, we will measure the accuracy all the time for each change that happens in the network. And for all the tasks, we will measure the accuracy. Now, the lowest accuracy we have for a specific task after learning it, that we will use for our minimum accuracy. And this is also what we average over the tasks we have learned before. And hence it gives some kind of worst case performance metric or stability component. So I hope this answers the question, but otherwise feel free to turn on your mic and elaborate.
I guess we'll move on to Vincenzo and Camila, and then we'll have a few minutes so we can come back to questions after all of the speakers have gone. So Vincenzo.
Okay, so I was, Matthias, can you hear me, right? I was wondering if you have, let's say, assessed this evaluation gap, performance gap, just by looking at the predictions of the model, or if you have also tested it somehow in terms of the representation layers. That is, I don't know, if something is happening at the representation layer, and then having like a kind of a linear evaluation would show the same gap in performance.
Yeah, so that's actually a very good question, and that's some of the follow-up work we're trying to look into, because now it's not clear on which component of the network indeed like the stability gap is happening. Is it really because of the classifier? But I mean, even if it's a classifier, we see it in these different scenarios: domain incremental, task incremental. Even if you have like this single classifier, it's happening. Or like in class incremental, it seems more intuitive because you introduce like these new class prototypes, right? That are basically random, and it makes sense that your problem becomes more difficult. You get lower accuracy. But even in this domain incremental scenarios, I mean, you keep the same output space and you're still observing this. It's very interesting. And it seems also to indicate it's also happening at the representation level. But something we still have to look further into, and indeed linear evaluation and so on would be really interesting to get some results in there. So thanks for your question.
And I think we will let Camila ask a question, and then we will let Camila talk.
Thanks. So thanks for the talk, Matthias. That was a super interesting presentation. My question is related to how can you deal with the situation, or how can you design an evaluation strategy for a situation where you have access to very little of the data seen in the past? Because, for instance, then the worst case performance, I would think, would improve a lot. So how can you deal with that?
So you mean the data you have available for evaluation?
Yeah, you have very few cases available for evaluation on previous tasks. So how can you still gauge sort of what would be the worst case performance?
Yeah, so I mean, it's a really good question, and it's also difficult in general, I think, to get a good estimation or evaluation if you only have a few samples of your previous task. So I think in continual learning, it's in general difficult to get a good estimation of the performance in general, and of course also therefore the worst case performance. We're also really inclined in continual learning to have, we have this really nicely aligned task sequences, and hence we can extract this evaluation data pretty easily, right? We have this held out data set. But in practical scenarios, we often have like really these data streams, and then it becomes also much more difficult to select even which data are we going to evaluate on, and how can we measure the different goals we're interested in for a continual learner. So yeah, it's a very good question, but I think it's very difficult if you have just a few data to, especially in continual learning, have a good estimation of your model.
So I think we'll wrap that up. Thank you again for that presentation. We'll make sure we let you have enough time to talk yourself, Camila, but please, as people have already started, you can continue the discussion also in the chat as well, and we can return to these after everyone has gone. So I will turn it over to you, Tyler.
Great. Thanks so much for the interesting talk. And today I'm excited to announce Dr. Camila González, who will be presenting on lifelong learning in the clinical open world. She just completed her PhD at the Technical University of Darmstadt in Germany. And we're very excited to hear what you have to say about the medical domain this morning. I think Matthias might need to stop sharing.
Yes, I would need to stop sharing. So thank you so much, Tyler, for that great presentation.
Matthias, if you're still on. Oh, cool. Okay.
Yeah, sorry.
No worries. Thanks.
Okay. All right, it should work.
Yes, wonderful.
All right, the floor is yours. We look forward to hearing it.
Thank you so much for inviting me. I also thank you so much, James, for being here so early, and whoever else is in a time zone that is not so, so pleasant right now. So thank you so much for joining. So I will talk a little bit about lifelong learning in the clinical open world, so for medical applications, and mainly for CT and MRT data. So why do we need continual learning in medical imaging? Well, actually, most of the research that is done on medical deep learning happens in static environments. So that means that even when we have data from multiple institutions or multiple scanners, we merge it together into one data set and divide it into training and testing. And this is something that we cannot do in a lot of real-world settings, because for once, we cannot often share data between institutions. We cannot move data out of the institution where it was acquired, and we sometimes lose access to data as time goes on due to patient privacy considerations. So we know that, of course, as Matthias also touched upon, deep neural networks tend to be unreliable in safety critical applications. This is partially due to the problem of shortcut learning, where deep models sort of learn the simplest solution that solves a problem. For instance, we have this example of a classifier for pneumonia that was actually only looking at these hospital-specific codes. We also have the wider problem of distribution shift, where slight changes in image properties cause models to deteriorate significantly in performance. And this is a very, very prevalent problem for CT and MRT data, because we have a lot of variations into how this data is acquired. This is based on the scanner used to take an image, also on the use and timing of contrast agents to highlight certain, for instance, blood. And we also have a lot of artifacts that happen during the acquisition. So here you see 4 different brain MR datasets, and you see here in the intensity histograms how different these are among each other. We also in parallel have changes in the patient population, also how diseases manifest and spread, etc. So what happens when we only design systems in static environments, where we have a lot of cases of disappointments in what we call software as a medical device? For instance, the system for detecting diabetic retinopathy. So here, what we try to do is tell when a clinician should take a second look to a patient. And when this was tested in a real setting, they were found that the quality of the images needed to be too high for the system to produce a good prediction. So this meant that nurses had to reacquire images very often, because caused very long waiting times, and it was overall a bad experience. Also, for COVID-19, we saw a lot of papers come out with systems for the automatic classification or segmentation of COVID-19 lesions, yet they had very little clinical utility, mostly due to a lack of external validation. You may be thinking, okay, this is still everything within a research context, but products that are commercially available will have gone through sufficient validation. However, what we find is that actually a big portion of the products that are now available for medical use cases have gone through very, very little external validation, and only a very, very few have actually gone through prospective clinical trials. So this means that the performance is actually evaluated in an actual real-world setting in a certain time frame. So of course, we can draw certain recommendations from this. We should always train with large heterogeneous data sets. We should strictly define what is the intended use and the intended conditions of a product and have more external validation. But as I believe all of us know who are here today, we have no guarantee that the performance will remain stable over time. And that's why we need lifelong learning, because real clinics are dynamic environments. All of these changes that I was discussing on the acquisition, on patient population, disease manifestation, all of these happen over time, and we cannot always predict which changes will happen. When this problem comes of data drift, our models deteriorate significantly. So what we want is to update models through their lifespan so that they can adapt to new environments. And also, of course, we want to deal with a situation where we may not have access to older data because of patient privacy considerations. So this is what we look for. Now, most of my research has been in domain incremental learning. So we have basically these different tasks, and the difference are due to mostly changes in the image acquisition and also in the patient population. So if we take a very brief look at the continual learning method landscape, we have regularization methods where we try to keep important parameters from changing. We have sparse connectivity approaches where we try to keep separate pathways for different tasks to prevent interference. And here we typically have the situation where we are able to prevent forgetting, but at the cost of certain plasticity in the model. We also have pseudo-rehearsal approaches, mostly based on the generation of data or knowledge distillation. And here we typically see more plasticity, but still a certain amount of forgetting. And we tested a lot of popular continual learning methods on the case of medical image segmentation. So here you see the segmentation of a prostate and the hippocampus on MR images. Here you see the ground truth segmentations for a test case, and then the performance of a model right after training with the first task that example belongs to. Here you see how the segmentation must deteriorate as we continue training, and how these different methods prevent this to a certain extent. Now, you see that none of them actually produce reliable segmentations as time goes on. If we look at rehearsal, then we see, okay, here we can still get quite a high quality segmentation, particularly for the hippocampus. However, in a lot of situations, as I was saying, we cannot rely on rehearsal because we don't have access to any of the older data that we saw. Another consideration when working with CTs and MRTs is that we typically have very small data sets with very high dimensional data. So we need to really minimize the use of GPU memory. Now, another class of methods are expansion-based methods. So here we typically add new trainable parameters, or we can also keep task-specific parameters and interchange these during inference. So in its simplest case, we would just keep different models. So a multi-model solution when we have one model per task, and just select the correct one during inference. Now, I actually think that considering the particularities of medical data, this is a very useful strategy. Now, if we look at sort of the simplest case of an expansion-based solution, we would basically start training a model. Then say that we have been training this model with images acquired with a Canon scanner, and now we start receiving images from a Siemens scanner. Then I would initialize a new model and keep training. And then during inference, if my image comes from a Canon scanner, then I would apply the first model state. Now, this becomes a little bit more tricky when we don't have information on task labels. So say these differences are not due to a different scanner or scanner manufacturer, but due to different acquisition parameters that may not be present in the metadata. So I always recommend, I always recommend comparing against such a multi-model baseline, because in such a multi-model solution, we don't have the problem of forgetting, and we also don't have any plasticity loss. So it's just for practical settings, I think it's always a fair comparison to make. Now, if we do have information on task identity, we can simply select the appropriate model state during inference. And if we don't have this information, then we can employ an oracle that basically allows me to select the most appropriate model state. And one method of doing this is XPerGate that you may be familiar with, where we basically train per task one autoencoder and then select the task with the lowest autoencoder reconstruction error for extracting my prediction. And this is something that we adapt for medical image segmentation by basically proposing to not keep a separate autoencoder based on a two-layer architecture, but actually keep the same architecture of the segmentation network, which also follows an encoder-decoder structure. Here you see the reconstructions that we have for 3 different anatomies with the in-domain autoencoder versus the out-of-domain autoencoder. So basically using this, we can select the appropriate model to use during inference. This becomes a little bit more complex, where we don't have basically these rigid boundaries between tasks. And this is actually a situation that happens a lot in clinical practice, because as I was saying, we have all of these different factors of change. We have acquisition parameters, reconstruction algorithms, we have changes in the population, the disease manifestation, and all of these are happening at the same time. So basically, there is not even a clear definition of what a task identity is. So how do we deal with this situation? Because then we have 2 additional challenges besides selecting the correct model during inference. We also need to detect with a sufficiently large shift in the distribution occurs and make sure that we keep our model pool small. So the way that we address this is by using the Mahalanobis distance in a low-dimensional latent space to detect these shifts in the distribution, which works quite well in a lot of medical imaging cases. Specifically, we go about this in this way. So we start training one certain model state. And then when a new batch of data comes in, we basically calculate the Mahalanobis distance to all of the states that we have. And if this distance is low enough, then we continue training a model, as you see here. And if this distance is too large, then this marks that I need to initialize a new model state. And then, of course, during inference, I would simply select the state with the lowest Mahalanobis distance to extract my prediction. Now, what we find is that by doing this, we don't have any plasticity loss, and yet we still preserve previous knowledge, as you see here. So this is our ODEX method in LILAC, and you see this compared against the elastic weight consolidation method, which tends to be a bit too rigid, and modeling the background. which is a bit too plastic. It's a pseudo-rehearsal approach. We also maintain the integrity of the segmentation. So here we see that even as time goes by, we can still extract quite a high quality segmentation because the model is still tuned well for that particular distribution. And a common criticism in multimodal solutions is that they require much higher computational overhead than other methods. This is actually not the case because, for instance, in this solution that I was showing, we are only updating or using one model at the time. So you see that we have minimally longer training times as regular sequential training, and we don't use any additional GPU memory. We do need more persistent memory to store these model states, but this is not really a constraint in practice, particularly in clinical scenarios where we actually have access to a lot of hardware, because as long as we keep the model pool small, so the number of models small, which is what we try to do with this approach, then it's not a problem if we keep a few more small parameter states. So this is basically a strategy that is quite practical and produces reliable results over time. We still, however, have certain practical challenges that hinder the use of lifelong learning solutions in clinical practice. The first one is a lack of unified evaluation standards, such as the ones that we have for, say, natural image classification, like split cipher, etc., all of the benchmarks that we are all familiar with. So trying to alleviate this problem, we worked on extending a state-of-the-art medical image segmentation pipeline for continual learning, where basically the users can very quickly try out an array of continual learning methods. Another challenge that we have in practice is that continual learning requires greater overhead by the clinicians on site. So for this, we recommend certain active learning techniques that reduce this overhead. We also have a suite with radiologists can extract saliency maps from the images, which is something that they really, really like to see. So these are just advancements to try to keep clinicians on board with continual learning. Now, the greatest challenge that we have right now when deploying such systems is that the current regulatory landscape doesn't allow for model updates unless they go through reapproval. Fortunately, there are a lot of initiatives that are trying to change this. So if we look at sort of how systems are approved today in the locked paradigm, the model would be trained, evaluated, approved, and then deployed. Now, under the new lifecycle regulatory approach, besides the documentation on the evaluation, the manufacturer would also have to submit documentation on the change specification, so what changes they plan to do. This would all be approved, and this would allow us to update the model as long as this goes according to these change specifications, and as long as we have real-world performance monitoring, transparency, and traceability. So, of course, this is only for changes in the model, not anything that would change the intended use of a product. So if we look again at a situation where we have an initial model that performs well, but then we have changes in the data distribution over time, and we compare these two paradigms, what we see is that in the locked framework, of course, this is what we have today, so it's easier to prepare documentation, etc. But we can only do certain updates in very infrequent intervals, because going through this reapproval process takes a lot of time and is very expensive, particularly now with the new medical device regulations. This can take up to 6 months. So we have long periods with no model updates. Now, under the new paradigm, I would still have discrete updates because we need to have this traceability, but this would be a lot more frequent, which means that overall my performance would be higher. This does require greater monitoring, traceability, transparency, but this is actually something that we want anyway. So to summarize, we have a lot of factors causing data drift in clinical environments, which cause deep neural networks to degrade. So in order to maintain good performance over time, we need lifelong learning, and we need to, of course, avoid catastrophic forgetting, as we know, without a loss in plasticity. And also, very importantly, try to not change the state-of-the-art architecture that we are using too much, because then often people would not try to prioritize continual learning over just using the architecture that achieves the best performance. Now, clinical environments are task agnostic, so we should try to not rely on rigid boundaries or knowledge on task labels. And we have these exciting new changes going on in the regulatory frameworks that we will see more on the next few years. So that was the presentation. Thank you very much for inviting me and listening.
Great. Thanks so much, Camila, for the great presentation. I think we have time for maybe a couple of questions, so maybe we can start with the one in the chat. So the question is, how large are existing public data sets that can simulate continual learning in medical data? For example, with different scanner manufacturers or different acquisition parameters. And then there's a follow-up that, do these data sets contain images from different hospitals?
Yes, thank you so much. That's a great question. So it depends a lot on the use case. So for instance, if we look at hippocampus that I showed a lot of results on, there are several public data sets on hippocampus segmentation. And this range typically, I would say from the smallest one would be maybe 30 images in total to around, I think, 125 subjects. So this is sort of the range that we are moving on. Then, for instance, for COVID-19, for lesion segmentation, the largest data set would be around 200 examples. So these are the ones for which the annotations are also publicly available. So this is basically the range on we are moving on. So in a lot of medical use cases, we have data sets that go from 20 subjects with annotations to maybe 200. Of course, we have some data sets which are larger, but that's basically the range we are moving on. And when we have basically one large data set, we typically have images acquired at different institutions, but very few information on which specific subjects comes from which institution, because this has all gone through anonymization. So due to anonymization, a lot of this information that allows us to do continual evaluations is lost. So what we typically do is to take data sets from different sources. So these were acquired at different institutions with different scanners, etc. But we don't have so much knowledge on all of the factors that are different between these different data sets. So that's a big problem right now.
Great, thanks. I see, Matthias, you have your hand up. You can unmute.
Yeah, awesome, thanks. So thanks for the great presentation. I thought it was really interesting to see how it's continual learning can be applied in the medical context. Now, you mentioned that replay, it's hard to use it in this medical context because of privacy concerns and so on. But if you have, for example, multiple clinics that you're working with, the knowledge that you learn from these different clinics, their data sets, is still incorporated in your network. And this is still like shared between the different clinics, and like this data is just like compressed within your network. How would you deal with that? So you can't use like the core images, but this knowledge is still somehow learned within your network.
So you mean basically the situation where we have, there are like different clinics, and I have used data from these different clinics for training the model. And you said, okay, we have this problem with replay. So actually, if we only consider the situation on this availability on where the data is located, we can actually also train with sort of federated learning protocols that keep the data in the different institutions, but still have access to this data. So this is something that we are working on, sort of these scenarios where, okay, we do have access to some of the previous data and we can do replay, but data needs to stay in the different institutions.
But at some point, data from these different institutions still consolidates within a single network, right?
Yeah.
So how do you deal with that then? Because you don't use like the raw images from all of them, but you still have the knowledge within your network that is still shared between these institutions.
I mean, that is expectation. I think so is your question about sort of regarding these privacy considerations, whether it's a problem that I keep this knowledge within the model?
Yeah, exactly.
Yeah, no, yeah, that's a great question that I get a lot. So it's basically about, okay, so how much knowledge is actually in the model? Does this break certain privacy restrictions, and we have this model that has been trained with these different data. Regarding that, there are actually no set standards on what it implies to, or how problematic it can be to keep this knowledge in the model. There are a lot of papers showing that private information can be regained from certain deep learning models, but there is not really a standard on how I can prove that such knowledge is not present in the model. So it's an interesting question, I would say. So basically in a practical, from a practical perspective, right now, you would not have the problem of having that knowledge within your deep learning model. You would basically, in the documentation, going through approval, in this case, it would probably be like in Europe, the GDPR that is restricting you and saving this information. You would just have to show in a certain way that the information cannot be regained, that would be traced, where I could trace back the subject. But there aren't really very clear standards on that. This is something that we need to work on.
Thanks.
Great. Thank you so much. I know we have a couple more questions in the chat. Maybe we can address those async or maybe in the discussion session after the main hour, but I want to make sure that our third speaker has enough time to discuss today. So thank you so much, Camila, and I'm going to hand it over to Andrea to introduce the third speaker.
Thank you.
Great. Thank you, Tyler. We're keeping this very dynamic today. We are constantly switching hosts, so be prepared for a last-minute survey on who introduced whom. Great. I guess we are perfectly seeing your slides. Thank you, Soroor. And I'm really glad to introduce Soroor Hediyeh-zadeh. She's going to present a talk on emerging topics in continual learning for computational biology. So we're kind of remaining in this biomedical domain. I'm very excited about that. She's a graduate student at the Computational Health Center of Helmholtz in Munich. So I look forward to your talk, and the floor is yours.
Thank you very much. Thank you very much for the opportunity to present. It's my great pleasure to be taking you through some of the works that we are doing at the intersection of continual learning and computational biology today. So RNA is a messenger that carries instructions from DNA to other parts of the cell for protein synthesis. And you might know that these are the proteins that ultimately regulate the biological processes in your body and are important for regulation of those for your health. So when it comes to when we want to explain to people how this RNA is measured, we tend to refer to this smoothie versus fruit salad analogy. So when we talk about single cell RNA sequencing, we mean that the identity of each of the cell from the mixture that we sample from the tissue is known to us, whereas in bulk RNA sequencing, we don't know the identity of the cell, but rather the signal that we measure is a mix, is a pool signal across different cells or cell types. And with single cell spatial, we mean that both the identity of the cell and the location of the cell in the tissue is known to us. When it comes to studying health and disease, we believe that it's typically one or more of these cell types that is involved in the aberration of the biological process or in disease progression. And so this is why single cell analysis and single cell technology is extremely insightful and important for us in biomedical research. I'll be covering two applications today, and these applications are concerned around modeling data acquired by bulk and single cell sequencing. So single cell RNA-seq is still expensive. Research groups can only generate a few samples. On the other hand, we have these larger scale public repositories, data repositories, such as the Human Cell Atlas, which is a comprehensive reference map of all human cells, and forms the basis of for understanding human health, for coming up with new treatments and therapies, for monitoring disease, and so on and so forth. And I really encourage you to have a look at this nice paper if you really want to find out about the full potentials that the Human Cell Atlas is bringing to the biomedical research. We also have data repositories such as the Cancer Genome Atlas, where we have RNA profiles sequenced by bulk RNA sequencing from probably millions of patients. And so almost always when we get a new data from a collaborator, we really want to study them and compare them in context of these existing larger scale data repositories. But there's almost always a distributional difference between public data and data generated by labs, as similar to scenarios that Camila explained and described. On the other hand, the complexity of the data that's being generated is increasing. So the human cell atlas, for example, mostly contains single cells from tissue, whereas our collaborators are generating single cells from cell lines or organoids, and these are biologically different. There are some inherent differences between them. And again, you can take it similar to the example that Camila mentioned, that population dynamic changes, perhaps the instrument changes. And in our case, it's actually both a biological and an instrument. Well, an instrument is the same, but it's mostly biological difference. So you can kind of say that these new data are from a different domain. And continual learning as a training scheme and as a field, that's its main goal is to prevent catastrophic forgetting and to model data that arrive sequentially and are non-IID, and where the distribution of the data changes over time, and even more importantly, as a discipline that accommodates learning scenarios that are task incremental, class incremental, and domain incremental, and so on and so forth, is really a promising field for us. So I'll be talking about two applications. And I really hope that by the end of the talk, I have convinced you that there are examples of unsupervised domain incremental learning scenarios or learning problems. In application 2, I'll also talk about continual disentanglement. So before I dive into application 1, these single cell RNA-seq data are typically processed into count matrices. So what we get from the instrument is a cell by gene count matrix, and then we model them through variational autoencoders, through deep generative models using variational autoencoders, where the generative process of the VAE is a probability distribution from the generalized linear models or GLMs. Why GLMs? That's because the gene expression count data often exhibit a very strong mean variance relationship. So negative binomial or zero inflated negative binomial are more appropriate statistical distributions. And practically what's happening in this variational autoencoder framework is that we learned the parameters of this of the negative binomial or distribution through amortized variational inference. We also get, from the latent representations, we also get cell embeddings. So you're looking at the UMAP of single cells. Each dot here is a cell. Each cluster is a cell type. And colors, different colors just distinguish different cell types. When a new data set comes in, we take this model that I described in A, and we just simply update it via transfer learning with a new data set. And this is really the state of the art. So when we receive a new data set from the collaborator, as I mentioned before, we want to say, I want to study that in context of the human cell atlas or one of the atlases in the human cell atlas using transfer learning. And that's important because then that enables me to identify cell states in my query data set. Or in the new data set that I get from the collaborator. But remember, as I mentioned, there's always distributional difference between query and the reference. And recently we realized that if the query is inherently different from the reference, i.e. it's from a different domain, transfer learning may work suboptimally. So in A, for example, you're seeing single cells from organoid in orange, and single cells from tissue in blue. And our reference cell atlas is these cells in blue, which are from the tissue. And as you can see, cells from the organoid, which are our query data set, don't really map. They do not really mix with the reference. And therefore, we aren't really able to say what cell states are represented in this query in these organoid models, single cells from the organoids. And indeed, when we look at statistical characteristics in single cell data and single cell organoid data, we realize that for some of the genes with high expression here, the gene expression variance is larger in organoid compared to single cell, which implies that perhaps even if the generative process, the generative distribution is the same, perhaps at least the parameters of the generative distributions should be different between query and reference. And whereas standard, the current transfer learning frameworks that exist in our field, they assume that they are from identical generative distributions. If we allow fine-tuning, on the other hand, we see that when we rehearse cells from the reference, the query model, we get very large reconstruction losses on the rehearsed cells, which indicates that forgetting happens. I think from the continual learning work, we aren't really surprised by that. And so these observations were collectively signaled us to look into continual learning. And indeed, when we compare integration with transfer learning to continual learning, we see that compared to transfer learning, we see better mixing of the cells from the query and the reference, i.e. orange and blue. And by the way, not all cells from these organoids should necessarily map, but to somewhere to the atlas. But that told us that overall we saw a better mixing. Cells from the query were closer to the cells in the reference, and we were able to recapitulate the biology. Overall, the message is that, it seems that with continual learning, we can get a better judgment and better assessment of cell states compared to transfer learning, which is a state of the art. And this is work in progress. In our work, we apply EWC and rehearsal. Despite the existing work, we found that, applying it might suffice to apply EWC only to part of the network. But similar to existing works in the supervised settings, we found that perhaps a simple even L2 regularization might be sufficient in unsupervised scenarios too. And consistent with the current works, we found that rehearsal really works best to prevent forgetting in VAEs. And but we know very well that these attempts are all to mitigate forgetting, to really accommodate the increasing complexity that we get in the data that we get. We need to also make changes to the generative model, and that's the work in progress. So perhaps the largest ever data sets that we have on RNA profiles from patients from a broad spectrum of cancer and disease, patients at different treatment stages, patients that survive, patients that don't, they come in bulk RNA sequencing. So bulk RNA-seq, this patient data, which is in bulk RNA-seq, is extremely it's an extremely useful opportunity. It's a huge opportunity for us to study cellular state changes over the course of the treatment or disease progression. However, if you recall the fruit salad versus a smoothie analogy, these cell states are practically lost in bulk RNA-seq. And so having a model that can bridge between single cell and bulk is extremely valuable to recover these lost cellular states. And we think that this is a domain incremental learning problem, and within each domain, there will be distributional shifts. And we think that techniques from generative reply and continual disentanglement can help here. Specifically, we model this, we train a DGM on our single cell atlas. We use generative reply to generate bulk profiles from the cells in the reference. Remember, our reference is typically tissue. And then we continue training on bulk RNA-seq from the patients using EWC or elastic weight constellation and replay. And what that means, and a schematic of this model is given in A, but that means that it means that we can come up with a joint embedding of patient profiles and tissue profiles for which we have the single cell measurements. And once they are on the same embedding, we can use a simple KNN to match each patient profile to the closest tissue profile. and for which we have the single cell measurements, and do a standard bioinformatics analysis to identify these cellular states that are different in the disease sample compared to healthy states. And since we are using continual learning, and the promise of continual learning is that we keep cell level representations, this is extremely helpful, particularly when we disentangle these representations at the bulk level with respect to the disease state, clinical attributes, or other perturbations, for example, response to the drug. With this disentanglement, we are able to, and having cell level representations, we are able to tell what are disease state specific changes in the cellular states. And so we are really keeping an eye on all the recent works that are coming out in disentanglement, and we think that's all very exciting. So here is an application of the model to a colorectal cancer recurrent tumor. So this is one single individual with a recurrent tumor. That means that the patient is treated, tumor goes away, but then comes back at after a while, and we want to know why, what happens. We found that by applying the model, we found that the majority of the cell states that seems to be present in this patient, in this single patient with a recurrent colorectal cancer tumor, for example, in the epithelial compartment, we were able to say to tell which cell types are more likely to be associated with the recurrence of the tumor. So in summary, I presented, so the biological problems that we look at in computational biology, at least for the ones where we use representation learning, are broadly categorized as unsupervised domain incremental. I covered 2 applications, and I showed that continual learning of genetic model improved upon existing methods or offered new ways. And also I briefly mentioned how the findings of the current CL benchmarks, continual learning benchmarks on non-biological data, were reproducible to biological data. Another exciting topics that I did not cover, I didn't cover today to keep the time, is the cell state shift modeling. This is where a population of the cells are transformed into another population upon response to a treatment or drug treatment. And we think techniques from continual causality and continual disentanglement would be helpful. So with that, I would like to thank everyone that enables my research, the TICE lab in particular, and thank you for your attention. I'm happy to take your questions.
Thank you, Soroor. That was a very interesting talk. And thank you to all the speakers. I guess we are right on time. I see, Camila, you have a question, right?
Yes, yeah, thanks for the great presentation. That was super interesting. So one of the questions I had was regarding the disentanglement. So you present one example of one factor that gives you quite I would say quite a meaningful disentanglement. So what are other factors related to disease or subject characteristics that you find helpful to disentangle?
Yeah, so at the moment, disentanglement is done really with respect to the disease state, because this is the largest axis of variation that we see in the data. But of course, we have other metadata about it. For example, as I mentioned earlier, if the patient responds, if for example, metastasis happens. It's possible, I think, as long as that factor of variation is continuous, it's discrete, we should be able to do the disentanglement. Having ways of disentangling data with respect to a continuous factor would also be extremely helpful, and this is what we are researching in. But in terms of prioritization, which we think which one is more important, I think that pretty much context dependent. And so far, it has been only on the disease state and disease phenotype, which is the major axis of variation.
Okay.
Thank you.
I think, well, I also have a question, but I think it's better if I save that for the final discussion. So I'll just hand it over to James for the final and general Q&A. So James, back to you.
Yeah, we'll go ahead and close this out, but then we're coming right back to questions if you have time. So I hope you can stay on, everybody. But I wanted to thank all three wonderful speakers again. I thought this was a great seminar, and I think so many of us have benefited from your insights. And we're going to ask you questions if you have time after this. But first, I want to remind everybody we are open to tweaking this format. So if you have any feedback or suggestions, please reach out to me. And we actually are going to say a quick word on the Continual AI Unconference. Tyler is going to mention that now.
Sure. Thanks, James. So if you don't use Twitter and you haven't heard yet, Continual AI is now hosting our very own unconference. So this will be coming in October. This is going to be a 24-hour long event that is virtual, so anybody can attend from anywhere. And we'll have a lot of different content. We'll have things like roundtables for people to discuss different topics within continual learning, hands-on sessions, mentoring sessions, some contributed talks that people can submit small proposals about giving a talk at the event. So maybe you're interested in doing that. One of the biggest components of what we're proposing here is this pre-registration process. So many other fields of science actively use pre-registration for submitting papers. Basically, as the timeline shows here, you'll conceive some type of idea, an interesting idea for continual learning that you'd like to work on. You'll submit a pre-registered proposal that's basically the experimental hypothesis you have and maybe a set of experiments and methods that you would like to investigate regarding that hypothesis. And then you'll go through a small stage of peer review. So basically, we'll have some expert reviewers in the field take a look at these different proposals and either accept or reject them. If your proposal is accepted, you will then have time to conduct the experiments that you outlined in your proposal. You'll have another round of peer review to make sure that the experiments are all sound and everything like that. And then your paper will be fully published in the PMLR proceedings. So within the next couple of months, some call for papers will go out about the pre-registration proposals. So if you have some ideas, maybe you can start thinking on those now. And the contributed talks call for talks will go out maybe sometime this summer. So you can also start thinking about that. We're very encouraging. So, if you have wild ideas, we want to hear them. If you have, if you want to give a talk that isn't just a paper and maybe it's a perspective that you have on the field, we're also interested in hearing that. So this is really a chance to throw all your ideas out there and have a safe zone for being able to discuss that. So yeah, we hope to see you at the conference, and I guess maybe we can share the link in the chat. I see Andrea already did that, but yeah, so awesome. We hope you'll attend, and we hope maybe you'll submit something for a talk or a pre-registration article as well. I guess I can kind of close this out. Yeah, so thank you to the three speakers that we had today. These were all really enjoyable talks. I think that it was a great discussion and everything. This has been recorded, so this will go up on YouTube soon, but we do like to maybe turn off the recording at this point and maybe have a longer discussion session for those who might not feel comfortable having things recorded and things. So thank you again, and we'll start the more ad hoc discussion session now.